plasmodium. These parasites cause the disease that we call malaria. Now, malaria is a tropical disease. This disease is actually transmitted by mosquitoes. These are organisms that when they invade the human's body, for example, right? They will invade the liver, and then they will invade the red blood cells. So when they invade the red blood cells, they tend to kill off the red blood cells. So the patient, when they are infected with these plasmodium parasites, with these red blood cells, they lose the red blood cells, so they cannot maintain the function of carrying oxygen in the body. And at that point, right, the patient will have very fever. But at the same, because they are losing the red blood cells, they feel very cold because they lose the ability to transport the oxygen. Okay, so in Vietnamese they call it. You know what is called in Vietnamese? High fever, but then the patient feel very cold, very chill. Yeah, that's right. So that's the Vietnamese version of the name, right? But actually, the name malaria actually come from the Italian language, because that during the time when the Roman Empire, you know the Roman Empire, like Caesars, right? Roman Empire, when they invaded the tropical country, especially in the Middle East, right? The soldiers died and they fell ill. And actually at that time, they didn't know why, because they didn't get anything, right? And they didn't realize that actually the disease has come from mosquito. But at that time, they blamed the air. They called malaria because they didn't know the reason of the disease. So they blamed the air. They call it bad air, man-daria. And that's how they had the name malaria. But of course, in Vietnamese, we call it sốt rét. So different meaning, right? So this is also caused by a kind of protozoa that I'm going to study in the next lessons. Another example is that, have you heard about the word red tide?
Yes, I haven't heard that.
Yeah, so you can have a direct translation, right?
Thủy triều đỏ, đúng không?
That's right. So actually, red tide is a kind of algal bloom. Algal bloom.
Tạo nở hoa.
That's correct. So actually, algal bloom are caused by different kinds of organisms, but there's one kind that causes the change of the color of the water into brownish or reddish that we call the red tides. Actually caused by the animal-like organism that also can photosynthesize, but they are brown in color, red-brown in color. And because of this, right, the algal bloom of this group, they have the name red tides when they develop so much. Okay, so these are the example. Another example of the protozoans are the tiny creatures that living in the guts of the termites. You know termites?
Yeah, they're quite annoying.
Why?
If you have a wood.
They are eating
eat wood.
Okay, so they're eating your doors, right? They're eating your doors or windows or even your
furniture,
right? The construction, because these termites are actually for all animals, they cannot digest cellulose. Okay, we cannot eat cellulose, right? Now we can eat cellulose, but we cannot digest cellulose, right? Even the cow, you know, the cows, they eat grass, right? They cannot digest cellulose by themselves. But thanks to the living organisms and the microorganisms in their intestine. For example, for the termites, right, in their intestine, there are bacteria that have to break down cellulose, and also the protozoans that help them to break down cellulose into sugar molecules. You know, cellulose is very strong molecule, right? It's not digestible by normal animal, but these microorganisms, including the protozoans in the gut of termites, help them to digest the wood and also cellulose. So these are some examples we can see, right? We have protozoans that can photosynthesize. We have protozoans that are parasitic to humans and animals, right? And we have others also can photosynthesize, but when they photosynthesize, they develop so much, they create trouble for the ecosystem, right? Then we also have the protozoans that are helpful, they are symbiotic for the termites, right? They are helping the termites to digest cellulose, right? So these are examples of protozoans, and they also function, their significance in the ecosystem and to humans' life. So exactly what are they? As I mentioned earlier, right? These are organisms that are single cells, unicellular. Okay, but not any single cell organisms. The single cell organisms that are eukaryotic. You know what eukaryotic means?
Sinh vật nhân thực.
Yeah, so they have the real nucleus, right? In the cells, right? So you're studying cell biology, you study mostly the eukaryotic cells. So you know the structure, right? And they are motile. What does it mean by motile? Yeah, so they can move around by themselves. Okay, they move around by themselves. Just like human, right? You can run, right? You can walk. So actually the word protozoans is derived from the Greek language. Proto means the first. Then we have the prototype, right? Of the things in industry now, they have prototype, right? So proto means the first. Zoa means animals. So in the Greek, literally they translate in the first animal because they have the first. They are the first group that have the characteristic of animals. Okay, so they are animalized organisms. So because exactly they are motile, right? As you mentioned, these are the key characteristic as well. Eukaryotic, as we already know, and unicellular. So these are the three things that you can see. Anything that have these three characteristics altogether should belong to protozoa. Because not all protista are protozoa. You have to pay attention to that, right? Not all unicellular organisms are protozoa. If they are prokaryotic, then they are bacteria. They are not protozoa. So these are the three characteristics that you need to pay attention to. Now, in the trees of life, right, protozoans can be placed at the base of the tree of life for animals, because scientists believe that some of the protozoans are actually the ancestor of the true animals. So that's the reason why we need to study protozoa in this course as well. We try to find the connection between the unicellular organisms, unicellular animal-like organisms, and the true animals. So just to put it in a bigger picture, you can see, right, not only unicellular eukaryotics are protozoans, right? They are collectively called protista. And in protista, there are three major groups, like fungus-like, they have algae, and they have protozoa. So you can see actually the position of the protozoas in the kingdom protista. Actually, protista is not really a fixed kingdom. There's still arguments whether we should keep the Protista is one group because they have three different major groups that are totally separate from each other. One is more related to fungus, mushroom, right? One is more related to plants, and the other one is related to animals that we are studying now. Okay, so where do they live? Right, you can see some example over there. Right, they can live on land, they can live in water, they can be out there in the environment, or they can be parasitic, like the malaria parasites we have just now, right? So they are also quite in, quite a number of types of environment on Earth. So there are several characteristics of protozoan that you need to pay attention to. The first thing is that they are, even though they are small size, because they are unicellular, but they have great diversity in terms of their body size and shape, right? As we mentioned earlier, right, they are eukaryotic and single cell, and they have the ability to move, right? So these are the key characteristics. Because they are single cells, so all the living activities are carrying out within oneself. So we need to also to look at their ability to uptake food and digest food within the cell. And they also have some specialized organelles. And these organelles only exist in some protozoa. Okay, and they do not exist in other groups. And also need to focus on their reproduction. So in this lesson and the next lessons, we are going to deal with this characteristic. Okay, so the first thing, now as we can see, right, how do they look like? Just now I already gave you some examples, but over here we can see more representatives of the protozoa. So they can come into any shape, right? This one doesn't have a fixed shape, the amoeba, right? Or this one look like banana, right? And this one look like a shoes, right? This one have even a straight-like shape with some kind of spines, right? This one look like sun, right? With all the rays, right? So they have great diversity in terms of shape of their cells, even though they are single cells, right? So, sorry. Further, when we look at the diversity of protozoans, then we can have a look. We can look back to this point again. Now, in general, because they're single cells, right? So most of them are very small. Of course, there are few amoeba that can grow up to centimeters, but this is very rare. So most of them are tiny. You have to measure them by micrometers, right? And the shape, as we mentioned earlier, right, this is very diverse. So either they have no symmetry. You know what symmetry means? Okay. So some group doesn't have symmetry, right? Or they have radiant symmetry. Or they have bilateral symmetry, but the bilateral symmetry is not very common in protozoa. You have a look here. So which one doesn't have a symmetry?
Heliozoa.
No, they have this one. They have radiant symmetry.
The amoebozoa.
Okay.
Are you asking asymmetry?
Yeah, symmetry. Asymmetry.
Asymmetry.
So amoebozoa and?
Yes.
Sorry, I don't know why it's keep running. Okay.
Dinoflagellata.
So they don't have a fixed dimension, right?
Yeah.
Symmetry. How about Euglenozoa and Celiophora?
They are like radial symmetry.
Actually, they do not have a symmetry. We come to that point later when we define symmetry. Okay? But basically, you can see that they are diverse. In size, in shape, right? In terms of the organization of their body. Now, the second thing is that they are single cell. With a nuclei, so like single cell and eukaryotic. So this is the basic structure of eukaryotic cells, right? The typical structure of eukaryotic cells. Okay, I have to check. So they have all the typical organelles in their body, like any other eukaryotic organisms, like mitochondria, the nucleus, right? The Golgi complex, the endoplasmic reticulum, right? All these are the structure that you can find in protozoa. So for this part, because you are studying cell biology, okay, so we are not going into details on this part. But this is so you need to bear in mind this is also one characteristic of the group. Now in the cell structure, right, I think in the textbook on page 12 to 19 also provide you a preview or overview, actually, an overview about the structure of eukaryotic cells in the textbook from page 12 to 19. Okay, so you can have a look at that, by especially focus on the cell membrane, cytoplasms, cytoskeleton, and on the typical organelles in the eukaryotic cells. Okay, so for this part, you can read at home. We spend more time on other things here. Now, when take a look at the cytoplasms, right, we'll take a look at the cytoskeleton, the contractile vacuole complex, and also the structure for their locomotion. Now, the cytoskeleton of the cell. What does it mean, cytoskeleton? Cyto means what? Cell, right? Skeleton means what?
Khung xương.
Okay, so cytoskeleton.
Khung xương tế bào.
Okay, so that's good. So basically, even in the, when you define, remember we define invertebrates, right? Invertebrates are the one with the backbones. But actually, you can see that organisms like invertebrates, they still do have bones, but they just don't have backbones. Okay, the bones here in the cells here, right? We call it cytoskeletons. Those are the structure. It depends on the group. It can be anything. It can be the protein filaments that make up the structure to keep the shape of the cells, right? Or the microtubule and vesicle. Now you can see here, in this one, right, everything seems to have like a fixed shape, except for the amoebozoa, right? Everything seems to have fixed shape. These fixed shapes are actually created and supported by the cytoskeleton. Okay, so even in the cell's membrane structure, they have the structure like vesicle. We're going to study the vesicle soon, right, to provide the support, right? So, so for example, over here, right, you can see some of the cells. They have some of this group. They have the structure beneath the cell membrane that we call alveoli. Okay, this is a system of membranous physical, and they just situated just beneath the cell membrane. This is one example, and it also provides the support for the cell. So they have kind of like extra buffer on the cell membrane. So the alveoli system over here is very similar to something that we have in our life. You know what? Have you ever looked at this before?
Yes.
And you know what you want to do with that, right?
To protect the fragile
Yes.
things.
But after that, what do you do? After one round, what do you do with this one? Are you playing with it?
Yes.
Yeah, you tend to like popping it, right? So
Yes.
the arrangement of these alveoli here is something similar to the bubble wrap. So you can see, right? In the membrane they have these small vesicles, right? Sorry. Okay. These structures, right? So it's something similar to bubble wrap. So it also create the buffer for the cells, and it provides support for the cells. It's not exactly like the bubble wrap, but they have similar function. Okay, so here's one example of showing you like you find the relationship between the form, the structure, and the function. So you can see in nature, right, living organisms, they already come up with the solution that we only invented like million years later or billion years later, right? So organisms already have this kind of structure in their body, even in the single cells organism like this. So it's something is very interesting for this group, right? So the skeleton is not like a bone, but instead they have the vesicle that we call alveoli beneath the membrane. Okay, so skeleton can be anything. Now, when you look at the cytoplasms of the protozoa, scientists realize that they are in the cytoplasms, right? It's not just simple, but actually they find that there are two parts. One is the outer layer, which is much thicker. You can see it here. It's very thick, right? They call it the gel phase or the solid phase. And the inner part that they call it endoplasms, right? In the liquid phase or solid phase on each part. Okay, and the interesting things about the cytoplasms of protozoans is that they can exchange the phase between endoplasms and ectoplasms. So the liquid phase can become the gel phase and vice versa. So you can see over here. And it create when they can alternate these things, they can create the movement that we call the amoeboid movement in protozoans, right? Even in certain cells of high animals, they will still maintain and still retain these mechanisms. Okay, we'll look at the locomotion later. We can have a look at the cytoplasm structure. Now, how do they move, right? So the structure over here was already give you some information about how they move, right? So, for example, the amoeboid movement will be something like this. Can you see it? Can you see it?
The video, yes,
Yes.
but it's not play yet.
It's playing now. Do you see it?
Yes.
Okay, so we can see the movement of the cells, right? So this is the amoeba, right? So you can see there's a flow of cytoplasms. Repeat it again. So there's a certain part of the flow, right? So the cytoplasms, the inner part is the liquid, so they flow into the arms or the legs of the amoeba, right? Sorry, hang. Sorry. Okay, so the cytoplasms, they can change the condition right between the liquid phase and the gel phase, right? So they can create the movement for the organisms, and that time we call it the amoeboid movement. And what does it mean in Vietnamese? Cytoplasms. Cytoplasms, right? It's actually that on the material within the cells, right? Surrounded by the membrane. Okay, so they also have other structure for locomotion, and depending on the group. They can either have cilia, which are very short. You can see the smear here in the picture. Right? These are the cilia, or the flagellums, or flagella in plural. So it's a very long structure. Now, another interesting things about this is, even though the size of the cilia and flagella are very different, one is like very small compared to the cell size, right? And the one is like much longer, right? But when we study the structure of the cilia and flagella, they're basically the same. The only difference is that is their size, right? So when they study the structure of the flagella and cilia, they have the same structure, which they have the part that we call exonyms. The exonyms are actually the core of the flagella. So part of the flagella and cilia are actually covered by the cell membrane, but within that they have the exonym structure created by microtubules protein, okay, enclosed by the cell membrane. Now, on top of the cell membrane, on this part, right, they found that there are microtubules arranged in a very unique and clear arrangement that can be formulated as two of the doublet microtubules and two central singlet microtubules. You can see over here at the cross section. You see it here? You see my laser point? See that? So you can see each round here is one microtubules. The doublets, so they have 9 doublets surrounding 2 central single microtubules or singlets. Okay, and that formula we call it the 9/2 + 2. Understand?
Yes.
Now, the interesting part is that both cilia and flagella have the same structure. And they also find this structure in higher animals. What does it mean? We find this structure in protozoans, and we find this structure also in true animals. What does that tell you?
That they were evolved from this.
That they are related to each other.
Yeah, that's what I mean.
Sorry?
That's what I mean.
Can you say again?
What I mean is they are related to each other, just like you told us.
Okay. So they're related to each other, right? So you can see the connection between some evidence about the relationship between the protozoans and the true animals. Now, if you look at the lower part of the structure of the cilia and flagella, right? It means beneath the cell's membrane or within the cytoplasms, right? The structure is a bit different, right, from the top. So the basal body, right? They call it the basal body. The pattern is different because they have triplets of microtubules and they don't have the central microtubule. So the formula for the base of the cilia and flagella is actually 9/3 plus 0, referring to the arrangement of the microtubules, right? As you can see over here, right? So this is like below the cell membrane, within the cytoplasm, right? So that they call it the base of the triplet. Now, this structure is the driving force for the most protozoa with cilia or flagella. They use this structure to create movement. Now, if you look at the cross section over here, you can see the doublets of microtubules are arranged in a certain order, and there's something sticking out here, like the arms, right? They call it the dining arms. The arms here are sticking to each other. And these arms, right, they can change the shape when ATP, you know ATP? You know ATP?
Yes.
What is it? Adenosine 3 phosphate, right? In cell biology, they call it the currency of the cells, right? Because the ATP carry energy, right? So when the ATP molecules stick to these arms, the dining arms of the flagella, right? They change the shape. So ATP attached to the arms of the microtubules, they change the shapes, and therefore they also change the shapes of the cilia and flagella. You understand that? Something like this, right? You can look at me. So for example, microtubules, right, they have the dining arms stick to each other like this, right, side by side, right? So when ATP attached to the dining arms, right, these arms change the shape. So they also create the slice between the microtubules and they bend the flagella. Okay? So when they bend the flagella, they create the movement. Understand it now? Do you understand it now?
Not really.
Not really. Okay,
Can you
explain
like
it.
explain it again?
Okay. Bằng tiếng Việt vậy nhá. Thì các bạn cứ tưởng tượng họ có trong mỗi một cái gọi là flagella hay là lông roi bơi đấy, đúng không? Hay là cilia là lông bơi đấy, đúng không? Thì cấu trúc giống hệt nhau. Cấu trúc 9x2+2 như mà nói lúc nãy, đúng không? Thì cứ mỗi một cái cặp đôi cái microtubules đấy thì nó đều có dư ra gọi là các cái tay dining arms, đúng không? Nó là các cái mẩu nhỏ đấy. Cái dining arms này nó bám vào các cái cặp microtubule gần nó. Các cái dining arms này có cái khả năng thay đổi trạng thái, tức là thay đổi hình dạng khi mà cái phân tử ATP, tức là phân tử mang năng lượng, đúng không, trong tế bào đính vào nó. Khi đính vào nó, truyền năng lượng cho nó thì nó sẽ chuyển đổi trạng thái, nó thay đổi cái hình dạng. Và khi thay đổi hình dạng dẫn đến cái việc là các cái sợi microtubule này nó trượt lên nhau. Các bạn tưởng tượng không ạ? Khi nó bám vào nhau, nó đẩy cái này, nó trượt này, nhìn kiểu tay tôi này, đúng không? Đúng không? Thì sẽ dẫn đến cái việc là khi nó trượt lên nhau như thế thì nó sẽ khiến cho cái lông bơi hay cái roi bơi nó sẽ bị cong đi theo những cái hướng nhất định. Và khi nó thay đổi cái trạng thái liên tục như vậy thì nó sẽ tạo ra được cái sự vận động. Đúng không? Cái câu hỏi khó hiện nay người ta chưa giải đáp được, tức là làm thế nào chúng có thể phối hợp các phần khác nhau của lông bơi, làm thế nào để mà chỗ này thì cong, thì cái này chúng ta vẫn chưa có cái giải đáp. Nhưng mà chúng ta biết rằng là nhờ cái phân tử ATP kết hợp với các cái tay lignin đấy ở trên các cái sợi microtubule, tức là các cái ống protein đấy, dạng trục đấy, mà nó sẽ tạo ra cái sự vận động của lông bơi và giòi bơi. Và đấy là cái cơ chế giống như chúng ta thấy là đấy là cái động cơ của nó. Rõ chưa? Đối với cái nhóm động vật nguyên sinh này chẳng hạn thì tùy theo nhóm, nếu như thì tùy theo cái dạng nó là lông bơi, giòi bơi, thì cách vận động của lông bơi, giòi bơi rất khác nhau. Ví dụ như đối với lông bơi chẳng hạn thì thường nó sẽ vận động theo cái kiểu giống như là hơi giống kiểu múa lụa ấy, các bạn biết không ạ? Nó tạo ra cái đội xoắn, tức là dạng giống như là gần như cánh quạt nó xoắn, nó tạo ra một cái lượng vận động, nó tạo ra cái hướng vận động phản lực. Còn các cái lông bơi thì là cách vận động nó hơi khác, nó uốn theo cái kiểu giống như mái chèo, nó tạo ra cái sự vận động khác nhau. Okay, so that's the mechanism. So depending on the group, for example, the group with the flagellum, they will create the propeller-like motion. So they push the organism in one direction depending on the way of the move flagella. But for the cilia, it's just like the row on a big boat, right? So there's also the coordination between these cilia, so they can move around by this kind of locomotion. So those organisms like protozoans with cilia or flagella, they move by this way, right? And if not, they move by, if they do not have either cilia or flagella, they move by their false leg or pseudopodia. I already mentioned previously, right? So they form the lobes on the cells, right? And thanks to the movement of the cytoplasms, right, they also can move. So these are another mechanisms for moving in protozoa. We call it amoeboid movement. Okay, so they're using this structure that we call pseudopodia, or in singular, pseudopodium, right? So they protrude outwards in the cytoplasms, especially endoplasms, right? Flow into these pseudopodia in certain direction. So that's another way for doing that, right? So either flagella and cilia or pseudopodia. Okay. So the structure I explained just now, right, about cytoplasms, right, is also explain the mechanisms how they move. Now the next question, because they're single cells, right? So they have to do everything within the cell, right? All the living processes have to be carried out within one single cell, right? So there's a lot of demands for many things, right? For locomotion, you know how they do it, right? And how do they, in a way, I call it, right? How do they pee, or how do they push out the unnecessary material from the cells, right? So for this part, so for example, in Coleophora, they have the structure that we call they have the structure called the contractile vacuole. You can see it here, right? Do you see the video? Okay, so you can see that there's something contracting here. So it keeps changing the shape, the size, right? From very big, brown, and become very small, right? The same here. So the name actually comes from that ability, right? So they can contract. Contract means they reduce the size, right? They contract. They contract for what? They contract to pump out excess water, to pump out unnecessary material in the cytoplasm, so they can maintain the volume of water, the volume of their cells, and also help them to maintain the concentration of material in the cells, in the cytoplasms. Right? So the contractile vacuole complex are actually common in protozoa, This is a specialized organelle. So usually they have the contractile vacuole and the vesicle tubules, right, that have to pump water out of the cells. And they have the function that we call osmoregulation. It means adjust the osmotic pressure or adjust the volume of water in the cell. The reason is that any living organisms, when they live, especially in water, there's always an imbalance between the concentration in the cell and the concentration outside the cells. For example, if they live in fresh water, right, the concentration of ions in the cell is always higher than the concentration in water in the environment. So there's more water in the environment, in the external environment, than in the cell. So you know the cell membrane is semi-permeable, right? So the cell membrane let water molecule goes in easily, isn't it? By osmotic pressure. So thanks to the structure like contractile vacuole, the cell can maintain the volume of water in the cells by pumping out excess water. Understand this point? Everyone?
Yes.
Okay, good. So this is the kind of structure. So when they have to do everything within the cells, right, their evolution will result in some structure, something like the contractile vacuole, that help them maintain the osmotic pressure in the cells. Okay, so these are the key points for today's lessons. Next week. So this week we already covered this point. The shape, right? Okay, so next week we will cover the two remaining points about how they take up food from the environment, and also about how they reproduce, and also their life cycles. That is about the contents about the next lessons. Okay, so I think the best way for you to do it, so you can learn it more effectively, right? Maybe at home, when you already have the PDF of the book, right? You look at the chapter about protozoa. Okay? Look at the chapter of the protozoas and look at the certain points here. Sorry. Look at this, right? To review. And so read a bit about how they take food and digest food in the cell. And so how they reproduce. Okay? That is for next week.